listening now? Anyone? Anish?
Yeah, sir.
Yes, sir.
Just I am again going to summarize this topic. Hello? Again, I am going to summarize. I have already delivered a lecture on the transaction today, and now let us understand the main points of the transaction. I am going to sum up. I am going to summarize. I am going to summarize the topic which I have taught today. Let us again be attentive to listen and listen me sincerely. And there may be some network problem by due to which you are not getting my voice there. Now be attentive and listen again. I am going to summarize the entire topic which have been taught today. Today I have taken up the topic of microbial genetics, which is the transduction, and I have already you have already studied in my previous lecture the transformation. I have already discussed with you and interacted with you on the topic transformation in my last lecture. Today I have taken the second method of the genetic recombination, which is transduction. Just I have said during my lecture that transduction is a virus mediated gene transfer from one strain of bacterium to another strain of bacterium. And in this case, there is no requirement of physical contact between the donor and recipient bacterial cell. Rather, a particular bacteriophage carries the gene from the donor bacterial strain to the recipient bacterial strain, and in this way the bacteriophage acts as mediator of gene transfer from the donor to the recipient cell. And you know that the bacteriophages are such viruses which attack a bacterial cell, and bacteriophages, as you know, that they are of two types: the temperate bacteriophages, and their life cycle is temperate bacteriophages, uh, and its life cycle is lysogenic, whereas the virulent bacteriophage, which life cycle is the lytic life cycle. The best example of temperate bacteriophage is the lambda phage, and the best example of virulent bacteriophage is the T4 bacteriophage. Let us understand the mechanism of this mechanism of transduction again. Mechanism, that is the process of how does the process of transduction take place. Let us understand it. At transduction is of two types: the generalized transduction and specialized transduction. In case of generalized transduction, any gene of the recipient bacterial cell is capable of undergoing transduction. On the other hand, in case of specialized transduction, which is also known as restricted transduction, a particular specific gene of the recipient bacterial cell is capable of undergoing transduction. And in this way, there are two types of transduction: generalized transduction and specialized transduction. Specialized transduction is also known as restricted transduction. The best example of the bacteriophage which brings about the specialized transduction is the lambda phage. The example of the bacteriophage which brings about the generalized transduction is P22 bacteriophage. Let us now understand the mechanism. What happens when a bacteriophage attacks the donor bacterial strain? Its genetic matter, its DNA replicates inside the host bacterial cell, and during the maturation or assembly period, a particular segment of the bacterial DNA gets integrated with the viral DNA. And when the bacterial cell undergoes lysis, the progeny phages are released outside, and some of the progeny phages may have the bacterial gene in addition to the viral DNA, and such bacteriophages which have the gene of host bacterial cell act as the transducing acts as a transducing bacteriophage. When this transducing transfer or aggressive bacterial cell. When the viral phage DNA is injected inside the recipient cell, the bacterial gene of donor strain get integrated into the genome of the recipient bacterial cell, and the recipient bacterial in this way gets transduced, and now we can say that the recipient bacterial cell has been transduced, and the bacteria which mediates the transduction is known as transducing phage, and transducing parts, and the transducer carries carries the gene from the donor bacteria cell have genes from the donor bacterial donor bacteria, and that gene has been carried from to the recipient cell with the this is the method of transduction. The any gene of the recipient bacteria is undergoing transduction, whereas in case of a specialized particular is gene can only get transduced. Therefore, this is known as the specialized trans. There are two types of transduction: specialized transduction